Good morning. Thank you, Janet and Bernie, for that introduction to what I think is going to be a very interesting conference, and especially now I think a very interesting introduction this morning, because I think it's important that we're all on the same page talking about what exactly regenerative medicine and cell therapy is. It's a fuzzy field in a sense because so many people think it's different things. So today we have a very distinguished panel who's going to navigate us through this. By way of introduction, I'm Dr. Max Gomez. I'm the medical correspondent for CBS in New York and have also been involved with stem cell conferences and therapies, et cetera, for a very long time. So let me begin by introducing our panel. First is Julie Allickson, PhD. She's a Chief Manufacturing Development Center Officer at the Wake Forest Institute for Regenerative Medicine. We have Colleen Delaney, MD, Founder, Chief Scientific Officer, and Executive Vice President of Research and Development for Deverra Therapeutics. She's also Professor, Department of Pediatrics, Division of Pediatric Hematology, Oncology at the University of Washington, and has gotten up very early to be with us this morning. And last but not least, Joanne Kurtzberg, MD. She is the Jerome Harris Distinguished Professor of Pediatrics, Professor of Pathology, Director of the Marcus Center for Cellular Cures, Director of the Pediatric Blood Marrow Transplant Program, Director of the Carolinas Cord Blood Bank, co-director of the Stem Cell Transplant Laboratory at Duke University Medical Center. She is also president of the Cord Blood Association. Does that all fit on one business card? Do you carry a whole purse full of cards around there? Anyway, so welcome and thank you all. Let's start with, I guess, really, let's do Regenerative Medicine 101. Joanne, since you've got the longest title here, what do we mean when we say regenerative medicine? That sounds like a pretty broad topic.
Well, you're right, Max. It is a broad topic, and I don't think it means the same thing to everyone who uses that phrase. But regenerative medicine to me means using biologics and cells and tissues to improve symptoms and sometimes cure diseases. And sometimes that means modulating the abnormalities causing the disease, like inflammation. Other times it means rebuilding organs or tissues that have been damaged by disease. So it's really used in a very broad sense.
And you said biologics, which is another sort of broad term that I'm not sure everybody understands. That's different from cells, right?
So cells can be biologics, but also products that cells make or tissues make can be isolated and turned into, if you will, drugs and used as biologics.
So Colleen, tell me a little bit about, let's talk about cells. What do we mean, or what kinds of cells we're talking about? Because again, everybody thinks of stem cells and that has been a very controversial area for a while there. Tell me what we're talking about when we're talking about cells.
So I think you bring up again a very good point, and a point that is often used in different ways by different people. I think the first thing is you brought up the word stem cells, and I think over my career, so I'm a stem cell transplant physician, so that's otherwise known as bone marrow transplant. So like Joanne, I have been trained in the use of blood-forming stem cells. So the issue there is that these are not embryonic stem cells frequently that we talk about here. Embryonic stem cells are one form of stem cells, but generally speaking, in regenerative medicine, cell therapies, when we talk about stem cells, we're usually talking about a more differentiated form, a more mature form of a stem cell. So, and cells themselves are just as it sounds. There are many different cell types in our body. So cellular therapy is not limited to the use of stem cells as that source. They really, I think of them, Joanne sort of hit it perfectly, but I think of cellular therapies or cells as living drugs, right? So this is how I often explain it to either patients or investors or people I'm talking with, that these are therapies that are living drugs. They're different than an aspirin that you get off the shelf, right? Which every aspirin is exactly the same. So there are challenges to this approach, but when we say cells, we really mean that. We mean the cells that are often derived either from the patient themselves or from another person, and they can be used to replace or replenish or fix diseased tissues or diseased issues within the patient. But these are living drugs.
So you bring up a good point there that Julie, let's, because being a living drug, cells though are really a little different than a drug. And what I've often heard discussed is, a drug may address one particular biochemical pathway in the body, whereas cells might be able to do a whole lot of different things, no?
Yes. So when you talk about a cell, I think the important advantage of it is that it can do a lot of different things by producing what we can say is growth factors, other factors that can create self-repair or can help to migrate other cells in that can help to repair. So they might be reducing an inflammatory response. They might be modulating or changing an immune response. And there's also possibilities of being able to be an antibacterial. So these cells, which is amazing, is it can home into the injury and it can help to repair. So that's something very different than the drugs that are chemically synthesized.
And can I add one thing? Sorry. I think what's really, as Julie said, is cool about this is that we tend to think of cells, and we have learned so much about the properties of these cells. So when we develop these cell therapies, our anticipation is that we're going to take advantage of what we know that cell will do in a normal body. So when we replace that cell into a patient, we expect it, as Julie said, to behave as it would normally. So if its job is to create or generate or produce a protein or something, we expect that that's what will happen. And that's what's really so cool about these things. And I think bone marrow transplant is the oldest form of cell therapy. And that really is that ultimate, right? We're taking these blood forming stem cells and miraculously we can give them to a patient, intravenously. They find their way to the bone marrow and they recreate the blood and immune system. So they then generate all of those cells for the rest of that patient's life. So I think we are harnessing and taking advantage of the science we know when we talk about each individual cell type.
And I just will add one more thing, which is that cells don't do just one thing. As Colleen said, a bone marrow or blood stem cell makes tons of other kinds of cells that rebuild the immune system and the blood, but cells are intelligent. And so they can react differently in different environments. And that's why they're exciting in regenerative medicine.
I want to follow on to say not only the things that we know, but also the things we don't know that they can do. So looking at how they actually function, we know there's probably other things that we don't know that they're helping with. So that will be discovered, but really important that it has several different mechanisms to repair and replace.
That's very exciting. And Joanne, you anticipated my question in that we can almost call cells, and I hate to use the term smart bombs, but they're smart, they're intelligent, and they'll do all sorts of things, some that we know that we want them to do, and some that, as Julie just said, might be doing some things that we haven't even figured out yet. So tell me, there are a lot of these different cells. You deal a lot, of course, with stem cell or cord blood transplant sort of things. What cells are really seem to be the most useful, the most available, the ones that can do the most at this point? Talk to me about different cells.
So, as you said, I've worked for many years with cord blood, which is a source of blood stem cells, but actually is also a source of cells that can be used to make immunotherapies and therapies that are active in neurologic diseases, because there are many different kinds of cells in cord blood. But there's a very exciting cell called an MSC. A lot of people say different meanings for those letters, but I call them mesenchymal stromal cells. And they can be derived from fat or from bone marrow or from cord tissue or from placenta, and probably other cells as well. And they are very exciting. They're being used in the clinic under several different clinical trials, and they modulate inflammation. And there are many, many diseases where inflammation is the main cause of damage to tissues and symptoms in patients. And the MSCs appear to be able to calm down those symptoms. And they're a very exciting kind of cell that's being explored in the clinic right now.
Yes, I think we're finding out that inflammation, or inappropriate inflammation, I guess, runaway inflammation, seems to be kind of the root of so many different disease processes in the body, right?
Right. Yeah.
So, Colleen, we're talking about cells, and I know you said that we're doing your background is in stem cell or bone marrow transplantation. And when we talk about putting cells into people, everyone thinks, well, we have to do a tissue match. We have to get it exactly or very close to the same type of tissue markers that exist in the recipient. But for MSCs, for example, that may not always be the case.
Yeah, that's true. And I think this can get very complex very quickly, right? So we also know you can take cells from the patient themselves, and that's called autologous.
Mm-hmm.
And then of course you're matched, right? Because they're your own cells and your immune system will not reject them. You can take cells from someone else, which is the term called allogeneic. But as you're saying, you can potentially use third-party donor cells. And those are cells that are literally sort of taken off the shelf, right? And that's the huge advantage of an approach like that, is that from a manufacturing perspective, because I think this is something that really needs to be emphasized, these are therapies that some of them have been FDA approved. There are many of them in the pipeline. They are scalable. They are going to be cost effective. So this is a very important part of that, but being able to use them, as you said, without tissue matching means they're available off the shelf, which means the development or the manufacturing or the production of these living drugs is so much simpler that way. So really the availability or the use of this type of product as a non-tissue typed or literally off the shelf type therapy really depends on what you want to use the cells for. So if you want these cells to live forever, like in a bone marrow transplant, literally become that person's new blood and immune system, they become a whole different, have a whole different person's blood system, those are generally have to be matched, right? Otherwise, at some point, they will not persist. But if you need more short-term fixes, if you need something to be temporarily present to overcome, as the example we've been using today, inflammation or inflammation gone wrong, to sort of reset the immune system or dampen that response, and then they can go away, then you have a whole, you open up a whole wide world of what these cells can be used for. And then they don't necessarily have to be matched specifically to a person. And that just enables a ton of different approaches.
That's really interesting. So, Julie, Colleen brought up manufacturing, and I know that is right in your bailiwick there. But so I want to ask you about manufacturing, but let me follow up a little bit with Colleen said. What is it about MSCs then that you don't have to match it? You would think you would, but is there some quality of these cells then that allows you to put them in, even if they're not going to last forever and ever, as in a stem cell transplant? What is it about these cells that make it so they're not immediately rejected? Julie, are you?
Sorry, I thought you were talking to Colleen. So the cells can come in just for a short amount of time, produce their growth factors and the soluble factors that they need to be able to heal or decrease or dampen that inflammation. And then they go away. That might be 5 days, it might be 2 weeks. So in that regard, they don't need to match. If you're replacing, or as Colleen said, in a bone marrow transplant, that's where you need that match. And those cells would then take over in place and stay for the longevity. So these other cells that we're talking about can simply go in, repair the damage or get the cells to migrate, your own cells to migrate to that area to be able to repair, and then they go away. So they do their work and then they're gone.
So short term, they're not going to elicit a big rejection.
Right. And
Right.
also the MSCs don't have the same, it's called antigens, but proteins on their surface that helps the body recognize foreign cells. So they're kind of disguised, incognito.
Cloak of invisibility.
Yeah.
That's exactly the way I've characterized it, the Harry Potter cloak of invisibility.
Yeah, and I think the other thing is we often take, in certain situations, we often take advantage of the fact that a patient might have a compromised immune system, right? So as Julie was saying, like when we put these cells in and they don't last, maybe it's days, maybe it's a couple of weeks, and sometimes that's a lot to be asked. If the person's immune system, which is really what's responsible for killing these cells, knocking them out, rejecting them, however you want to say that, if the immune system is intact, those cells don't last long. But often if someone's had, say, chemotherapy, or if their bone marrow or their immune system has been damaged by something, they don't have that same ability. So we as, physician scientists can take advantage of that knowledge and infuse these cells at a time where they can persist and hopefully repair that patient's immune system. And when that happens, the cells get rejected because now you have a healthy immune system again.
So Julie, what we've been talking about here a little bit is about putting these cells, MSCs, into the body and letting them sort of figure out in a way what needs to be done. But I know that at the Wake Forest Institute for Regenerative Medicine, you're using these cells to actually build biological structures in organs. How does that work?
Yes. So I think a lot of you know that, back in the early 2000s, Dr. Tony Atala had the first lab-grown bladder that was implanted into patients. And so we're fortunate enough that he brought a lot of that technology as well as all the development that takes place at Wake Forest. But we work with different organs and tissues using a scaffold or a structure, and that could be synthetic. So it could be produced, or it could be a tissue that's biologic in nature that we've taken all the cells out. We typically then take a biopsy from a patient, tease out those cells and expand them and reseed that structure or that organ so that we can implant that into a patient. So I'll just use the example of tissue engineered urethra. That's a synthetic scaffold made out of a material that we seed with the patient's cells, and then it matures in a bioreactor, it incubates before we implant it into a patient. So if there's a damage where we have to repair part of the urethra, we can use this technology to be able to do so.
And
so
that scaffold degrades or is reabsorbed over time?
Yes, so that's a great question because a lot of people think, okay, you're replacing it. So just so you know, that does degrade over time. And it's like, I'm going to make it analogous to a good marriage. It's degrading, but then you're producing the native tissue at the same time. And so you end up with a native tissue of your own because your cells migrate in and help to form that tissue. So it's just there temporarily to produce a structure so that your body can actually do the repair. So it's pretty amazing how it works.
And in some cases, you're actually using sort of highly modified and adapted inkjet printers, bioprinters, to actually make mini organs?
Yes. So there's a lot of work and discovery right now at the Institute. Some of the things that you're talking about that we do, we also look at what's called organoid, a small organ. And that not only could be looked at in the future to be able to repair for infusion, but also to test drug development and really understand how the organ functions. So there's a lot of opportunities for that. On the other side, we're also looking at 3D printing to be able to print organs for future clinical work, as well as looking at printing directly onto a patient where there's a burn. There's a prototype of that at the Institute. So there's a lot of different angles we look at for 3D bioprinting, and that's definitely future thinking, because there's nothing in clinic right now with bioprinting. But if you could think about it, how amazing it would be, because you'd be able to centralize all of that technology, where possibly you'd be able to produce the organ on site to implant to the patient. So that's what we look forward to.
Amazing. So Joanne, you wear a lot of different hats in the cell and regenerative therapy field. How would you characterize sort of the state of the science, and how do we move it forward to actually get it into people, into use, into the clinic?
So I first have to say that I think this is an area of enormous potential. I think it's going to be the next big development in medicine. Not that we've exhausted drugs, but we've exhausted definitely the low-hanging fruit using drugs, and cells, because they're intelligent, and tissues, for the same reason, have much more capabilities. I think the field right now is really still in its infancy. There's great signs of promise, of efficacy, and there's a lot of data, particularly for the MSCs, about safety, which is all very favorable. But we really need an investment in clinical trials that can be done properly and can figure out where these cells work, how they work, what diseases they work in, because it's not going to be one size fits all. There's a huge opportunity, but it's going to require an investment, I would say, from government, from industry, to really figure out which cell works for what, and how to best manufacture it, and how to upscale that manufacturing, and how to make the cells available for large numbers of patients.
So Colleen, one of the areas that everybody is very excited about is some version of using cells for cancer therapy. And I know you've been working on what is benignly called NK cells, but it stands for natural killer cells. Tell me what those are and where they come from, and how would you use a killer cell?
Well, first of all, what a great name, right? Who doesn't want to be a natural killer against cancer? So natural killer cells, or NK cells, are really, I like to describe them as our body's first responders against transformed cells. And so whether that cell is transformed because of cancer, or whether that cell is transformed because it has a viral infection, right? So it's been virally infected. Our natural killer cells, without training, right? So they don't have to be educated against that specific, say, target. These cells can hunt and kill transformed cells. So they really are your first responder. And again, this kind of goes back to what we were talking about, of taking advantage of the advancement in the knowledge of how certain immune cells work and using them to generate, hopefully, curative therapies. And as you know, Julia and Joanne have talked about, I too believe that these therapies are the wave of the future, and they're going to be able to be given without tremendous side effects. Natural killer cells are very exciting because we can use them off the shelf, like we were talking about before, which enables us to make them ahead of time. The cells that we work with, we derive from cord blood stem cells, blood-forming stem cells, and we've effectively recapitulated the bone marrow in the lab, and we expand and number these stem cells, and then we direct them to do as they would in your body, and one of the cells we make are natural killer cells. We can then test them, their potency and their ability to kill cancer targets. The other really cool thing about these cells is that they can be engineered, and we haven't talked a lot about gene therapy, and I don't want to confuse things, but effectively you can even target these cells even more than they already are as a natural source. And then you can really hopefully increase their efficacy by using smaller numbers of them. They can target the cancer cell you want them to target. They don't, have any other side effects. So the idea is that you can generate these products, you can put them in the freezer, you can find a patient, and hopefully someday we're really going to have very personalized approaches with this. And you can infuse those cells into a patient with cancer or with an infection, and they can hopefully help replenish the body's immune system to fight that cancer or that infection. So at some point you might ask, well, if natural killer cells are our body's first responder, if you get cancer, they've clearly failed, right? Because now you have cancer. So at some point, the body does get pretty smart. The cancer gets smart. It puts on its own invisibility cloak. So even though those NK cells are still there and maybe functional, they get tired or they can't see the cancer anymore. And that's where this idea of using a living drug to replenish and to revitalize that immune system to effectively kill the cancer is important.
So these NK cells are the equivalent of calling in the cavalry to help the killer cells that we naturally have. We're bringing in reinforcements.
Right. Kind of like we're doing out here on the West Coast with our fires.
Oh boy, I hope you get plenty of reinforcements.
Just need rain.
You need the hurricane to come there.
Yeah, exactly. So I see that we're running a little bit low on time here, and I've been threatened not to run over time here. And what I like to do at the end of these sorts of panels is ask our panelists for a take-home message, a pearl of wisdom here, so that our listeners can remember something to take home from all of this. And I've forewarned our panelists, so they should be prepared to have a little brief pearl of wisdom for us. And Joanne, since I know that you have so many different hats to wear, pick one and give me a little take-home message, please.
My message is that cells and tissues are incredibly exciting sources of novel therapies that can be developed to treat unmet medical needs, and that we need investment in those areas to be able to realize their promise.
Exactly. Colleen, you're up.
Well, see, we didn't talk about what we were going to say ahead of time. So I would absolutely agree with Joanne. That's, I think, the biggest pearl. But I will add to that and say that the other area, so first of all, this is tremendously exciting and the future is incredibly bright with these therapies in them. But I would add that we really need to think about this. We need to anticipate the success and the ongoing approval of some of these therapies. And along with that, we need to increase the training of the workforce that will enable these types of therapies. So we need to be smart about this. We need to interact more with the FDA. We need more centers that will be able to generate and manufacture, like Julie, manufacture these cell therapies. And we need to think about smart clinical trial designs and reducing cost of clinical grade reagents. Excellent.
And in a way, I guess, the future is now. We have the future, but the future is also now. It's already happening. So, Julie, you're taking these cells and as you said, that Dr. Atala has already used some in people. What's our take-home message for using cells for manufacturing?
Just that there is so many advantages with the cells, and Colleen mentioned a lot of things, but we need to be able to look at sources, raw materials, and things that help the manufacturing to be friendly. And FDA has really helped us with several paths that make us efficient. So I would say, speak to them early. And at the end, I really want to say that I think that these cells can increase the quality of life. And there's even evidence through studies that it can decrease the healthcare costs. And we all know right now, healthcare costs are at about $3.7 trillion per year. And we really do need that. I think everybody needs to think about that. And if we can get the funding in to push this and get it going, I think we can get the healthcare costs down. That's really my hope.
That's terrific. So I want to thank our panelists. I think that you all have done a terrific job of laying the groundwork for the rest of the conference today. And now I hope that everyone understands a little bit more about cell therapy, MSCs, regenerative medicine, and how important this entire field is, both for investment and continued research. So thank you all. Have a great rest of the conference, and we will see you in the cellular therapy world.
Thank you
so much.